The milestones in the study of luciferin showcases the curiosity and contributions made from thinkers across the centuries. These explorations stand as a testament of enduring the power of scientific discovery.
How can such a common chemical reaction create such an unfathomable and breathtaking sight, lighting up the night?
From the smallest insects that walk on land to the biggest predators of the deep sea, luciferin can be seen at work. Its applications and uses endless.
Luciferin's luminescent properties illuminate medicinal pathways for non-invasive imaging and innovative diagnostics.
We can start by talking about the history of luciferin. In 1667, Robert Boyle calculated the air required for luminescence. At the time, oxygen had not been discovered, but we know now that it was an oxygen requirement for the oxidation of luciferins. Boyle also found that the intensity of the light depends on the amount of oxygen present. During the 19th century, Raphael Dubois used Boyle's findings to experiment with bioluminescent clams and cold water to make a paste. He boiled half the paste and the glow stopped. Once it cooled, it was mixed with the unheated sample and the light was emitted again. This observation led to Dubois' discovery of a heat-stable organic molecule named luciferin. In 1953, William McElroy and colleagues found that in bacterioluminescence, luciferase catalyzes a reaction of a reduced form of luciferin called flavin mononucleotide. McElroy had stated that they repeated Dubois's old experiments to see what he had found, and it turned out he had found ATP without even knowing. I'm going to pass it off to Katie to talk more about the chemistry.
Let's discuss the chemistry behind luciferin. Luciferin will undergo an oxidation reaction driven by ATP, where it will yield oxyluciferin as well as several other products, including carbon dioxide and the light that we see, which will create the bioluminescence. Luciferase is the enzyme that catalyzes this reaction, where the substrate, luciferin, will bind to the active site of luciferase with its cofactor, Mg2+, to catalyze the oxidation. It is important to note that there are several analogs of oxyluciferin, including the yellow natural one at 568 nanometers that we see emitted from fireflies, as well as other synthetic analogs that have a variety of practical applications. So what happens exactly? Well, when luciferin binds to luciferase, its enzyme, in the presence of ATP and Mg2+, as well as in the presence of oxygen, it will be oxidated. This will yield pyrophosphate, as well as its intermediate, luciferyl adenylate. Luciferyl adenylate will be oxidized subsequently, resulting in AMP being released, as well as the keto-enol form of luciferase, due to the fourth hydrogen being removed from the carbon ring. From the keto-enol intermediate, carbon dioxide will be released. This will yield the excited state of oxyluciferin. The light energy we see, the bioluminescence, is a result of the excited state of oxyluciferin resolving to its ground state, which will release a photon of light, producing the bioluminescence in fireflies and other organisms.
Okay, so a couple of organisms can glow in the dark, but why does this matter? Well, it turns out this luciferin-based bioluminescent light has proven to be both significant to us humans and the organisms from where we extract these proteins from. Within larger organisms that create bioluminescence, most of them follow one of two structures. They either possess bacteria within itself or on its surface that follow the luciferin-luciferase pathway, or they possess photophores, which are glowing organs that utilize lenses, reflecting layers, and filters in addition to the light-producing material. Bioluminescent functions typically fall between these three categories of offensive, defensive, and communicative functions, and are often found overlapping across these categories depending on the organism that exhibits the function. For example, we now know how fireflies create this bioluminescent light, and you probably already know that they do so in order to attract possible mates. But did you know that glow patterns differ across firefly species as well? And as a result, certain fireflies, such as the female Photorus, will actually mimic the glow pattern of other species in order to lure them as prey. Another example of cross-category function would be that of counter-illumination. So since predatory species typically like to hunt from below within the water, many organisms will excite the luciferin photophores on their underside and manipulate the wavelength and light intensity to match the overhead natural lighting that's produced produced by the sun or the moon for nocturnal species. This overall reduces the contrast between the organism's silhouette and the background as seen from below. Predatory species will also utilize this technique. The cookie-cutter shark will illuminate its entire body but purposefully leave a small patch near its pectoral fin unlit in order to mimic that of a small fish in order to lure potential prey. Even dinoflagellates and copepods have what's known as a burglar alarm system. They both emit bright light when threatened in order to attract predators of a higher trophic system in order to endanger their initial threat. This also prevents them from being eaten, as their glow can still be seen within the stomach lining of many organisms. Concoctions of bioluminescent chemical mixtures and bacterial slurries are expelled like ink in certain squid species in order to daze and stun predators. Some squid species will even illuminate their own limbs and then amputate them off in order to deter and confuse their predators as well. The functions go on, and luciferin-based bioluminescence is only as limited as how deep the ocean is, along with the organisms that live down there. But enough about organisms. How have humans found a way to utilize this interesting phenomenon that's found in nature?
So researchers have actually found many ways to utilize bioluminescence. So as a little background on the application processes, the different luciferase pairs have different light emission wavelengths and are reportedly suitable for different applications. And the last decade or so has seen great advances, which have allowed luciferins and luciferases to be used in different ways. So some of the key traits are the sensitivity, the resolution, and the selectivity, whereas other optical imaging agents face obstacles such as being overly large or polar, which hinders their effectiveness. So what has developed within the field of research, this has led to luciferin playing an important role in bioluminescent in vivo imaging, utilizing this natural light production to allow for the generation of non-invasive whole body pictures of living organisms. So some notable applications of bioluminescence include the labeling of cells and more. And this process actually allows for the monitoring and detection of possible tumors within living organisms, as well as aiding in analyzing the effectiveness of drug responses. So recently, this was used for monitoring the growth of brain tumors in mice. So what happens is that the gene encodes for firefly luciferase gets introduced as a reporter gene into the mouse, and then that mouse is then placed into the light type box. And the photons are released when the luciferase oxidizes the luciferin, and they're detected using a charge-coupled device camera that localizes the photons. So colored images that represent the signal intensity are overlaid with the grayscale reference images of the animal to show the location of the signal. And this allows scientists to monitor the growth of the tumor as well as the response to its treatment. And because the imaging is highly sensitive, it can detect very small concentrations, which combined with the fact that firefly luciferase has a fast turnover, this allows for real-time measurements to be made. So this has also been used to analyze pathogens and their effects within the human body. So one area that's especially relevant is that the infectivity of coronavirus was determined in different host cells using a FLUC and D-luciferin assay. And a similar assay was also used to establish that SARS-CoV-2 and RATG13 coronavirus that was found in bats can both successfully infect human cells to produce daughter viruses. So as pictured, the pseudoviruses would have the desired spike protein on the virus and on their surface, as well as the genetic material encoding for the bioluminescence inside them. And after being incubated with ACE2 expressing human cells, if the viruses are successfully able to infect the human cells, the daughter viruses with the bioluminescence would be produced. And with D-luciferin, the light output would be able to measure the infectivity of the cell.